Thanks very much for the last session. I'm Stephen O'Leary. I'm a fellow. I'm a surgeon scientist with an interest in ear health at the University of Melbourne, here in Melbourne. So, look, it's my great pleasure to chair what I think are probably the most exciting parts of the program, and that is to hear from some of the new fellows. And I think all new fellows are very, very welcome, but not everyone, unfortunately, can get to speak. And I still remember when I was given that opportunity. So I'm really keen to hear from everyone. Everyone has 15 minutes, of which they've been asked to speak for 10. So that we'd like some robust dialogue and questions online and with the roving microphones. If the speakers decide to use their 15 minutes up, they don't get questions, but we don't want that, okay? So look, with no further ado, I'd like to introduce our first speaker, who's Professor Rajaratnam, who I noticed gave a question a little bit earlier. He is the academic head of the School of Psychological Sciences and Deputy Director of the Turner Institute for Brain and Mental Health, Monash University. Thank you.
Thank you very much. And I'd like to begin by acknowledging the traditional custodians of the land on which we meet, the people of the Wurundjeri nations, and I pay my respects to elders past, present and emerging, and also extend that acknowledgement to First Nations people who may be here today. So I was asked to speak about a story of some work that I have been doing that summarises at least an aspect of the research program that I lead. And so I've decided to tell you about the story of melatonin and melatonin agonists and how they became used in the treatment of circadian rhythm sleep-wake disorders. So circadian rhythms and their implications for health have been increasingly understood in the past few decades. We now know that there are multiple clock systems, both a central clock system in the brain, but also distributed throughout the body. And we know that disruption of these clock systems result in multiple risks to our health, and those are summarized on the right-hand side. And we know that there are particular disorders in which this disruption occurs frequently. And so we know in shift workers, for example, who have a misalignment between the internal circadian timekeeping systems, the sleep-wake cycle, behavioral cycles, they are particularly vulnerable to a number of these conditions. The bottom example is common in blind people who lack light input, and they suffer commonly from a disorder called non-24-hour sleep-wake disorder. And so this gives you an idea of how the physiology of the system is now increasingly understood. Now, one of the reasons I chose to talk about melatonin is because next year marks the 40th anniversary of the first publication that melatonin is able to affect the mammalian circadian system. And this discovery came from none other than La Trobe University in Australia, and it completely revolutionised the field of circadian medicine because it offered the first pharmacological treatment for circadian rhythm sleep-wake disorders. And so this was work published in Science by my former PhD supervisor, Jenny Redman, and she showed in these elegant experiments that daily injections of melatonin can entrain the locomotor activity rhythm of rats held in constant darkness. And this was really pioneering because until that time, it was thought that the mammalian circadian system could only be influenced by light. Now, I extended this work from a nocturnal rodent in my PhD to start looking at a diurnal rodent. And so I got these diurnal rodents from Western Australia in Perth, around Perth Zoo, where they were introduced from India. And I did these experiments at Monash University. And so we followed a similar protocol to what Jenny Redman and others had been doing in rats, where we looked at whether a diurnal, day-active rodent was also responsive to melatonin. And indeed, we found similar synchronizing or entraining properties of exogenous melatonin. I then went on in my postdoctoral research at the University of Surrey in the UK to extend this into human proof of concept studies. And so working in a Ministry of Defence and MRC funded study, we looked at whether a dose of melatonin, 1.5 milligrams, could shift the timing of the circadian clock in humans and also directly influence sleep. And we found indeed that it could, and it had powerful effects on sleep as well as the circadian clock. And just to illustrate how we represent sleep in this kind of pictorial format, the colour contour plot indicates the intensity of sleep or the efficiency of sleep, with the red and orange being high efficiency and the blue and purple being poor sleep. And so we gave these people on consecutive days of the study either placebo or melatonin, and I'll show you the melatonin in a moment. Now, they were put to bed at 4 o'clock in the afternoon, and as expected, for the first half of this extended sleep episode, there was very little sleep. But then in the second half, from about midnight onwards, we see fairly strong consolidated sleep. Now, when we gave melatonin every day in a randomized double-blind fashion, we found that the distribution of sleep was completely altered, where sleep was now occurring much earlier in the sleep episode, so the distribution was completely changed. We measured markers of the circadian clock, melatonin, cortisol, and pituitary hormones and so on, and found that all of these were shifted. So I then took this work to the United States, where I worked at the Brigham and Women's Hospital. And I led some trials of a melatonin agonist for the treatment of circadian rhythm sleep-wake disorders. And this particular melatonin agonist is called tasimelteon. And so we first did a phase 2 trial and then a phase 3 trial where in the phase 2 trial, it was proof of concept, similar to the work that I did with melatonin, but this time 3 days in a kind of a jet lag protocol, where 3 days of exposure to the randomized treatment. And what we found was, again, here's the placebo condition. On the first day in which you advanced the sleep-wake cycle, you can see disruption of sleep that occurs for the first few days. And this is the typical kind of jet lag paradigm, but it was done in the hospital inpatient facility. And with increasing doses of the melatonin agonist, you can see that on the first day in which the sleep-wake cycle is shifted, particularly at the highest dose, we see sleep is restored to the baseline level. Now, the trials continued in non-24-hour sleep-wake disorder, and this resulted eventually in the FDA approval of tasimelteon as the first circadian regulator for the treatment of circadian rhythm sleep-wake disorders. Coming back to Australia, we decided to then take this work forward with melatonin, because one of the common criticisms I got was that, well, this is an expensive drug, melatonin is freely available, can we do this with melatonin? So fortunately, we convinced the NHMRC, and the NHMRC funded a randomised controlled trial across 3 sites, at Flinders, at Monash, and at the University of Sydney at the Woolcock Institute, and the study was led by Tracy Sletton. And so what we found in a patient group called a delayed sleep-wake phase disorder group, which is a disorder of the circadian timing system that's common in adolescents, we found that a 0.5 milligram dose of melatonin significantly improved both patient-reported sleep outcomes as well as objective sleep outcomes, and also clinician-assessed outcomes, including the global improvement scale. So this provided us with really robust evidence that the naturally occurring hormone melatonin is able to significantly improve the sleep and other outcomes of these patients. More recently, we've looked at predictors of treatment response to melatonin, and one of the avenues that we pursued is looking at the increasing number of circadian clock genes that have been identified and are known to be involved not only in the regulation of the circadian cycle, but we know that polymorphisms of these genes are or attributed to some of the circadian rhythm sleep disorders. So we looked at one of these polymorphisms, which is in one of the circadian genes per3, and indeed we found there were substantial differences in treatment response depending on the genetic or the genotype of the individual. So I've been asked to keep strictly to 10 minutes, so I'm going to stop there. Thank you again to the Academy for inviting me to speak today, and I thank all of my colleagues who have been involved in this work at Monash University, at the Brigham and Women's Hospital, Harvard Medical School, and the other institutions throughout Australia. Thank you.
Well, thank you. I hope there are some questions from the floor. Just a comment, you know, it's interesting that for me in ears, melatonin is one of those things that protects the inner ear. So it's an interesting idea, I think, that melatonin not only regulates sleep where a lot of your body's repaired, but it actually can protect the sensory systems as well. Can you comment on that?
We have considered melatonin as, I mean, there are lots of direct effects on the systems as well. We've been thinking about melatonin as a regulator of processes that occur during sleep. So we thought at one time that sleep was an inactive process, but we know now, as you say, that it's an active process where lots of repair and recovery occurs. And we think that melatonin is involved in that signalling of that timing.
Right. Excellent. Any other questions?
Sue Davis, Melbourne. Thanks for that talk. I've got 3 very short questions. One, does it affect other clock genes other than the central clock? 2, you were talking about very small doses like 0.5 per milligram, but what you get over the counter in, say, the US is 5 to 10 milligrams, and the people being overtreated. And thirdly, do people build up resistance to treatment?
Great question. So the first one, we haven't looked at the direct effects on the expression of the clock genes, either centrally or peripherally. What we looked at there was naturally occurring polymorphisms of those genes and looked at differences in treatment response. But we haven't looked at the acute effects because we know, for example, light has acute effects on the expression of those genes. Your second question about the dose is a really important one because when I consulted probably the premier pharmacologists that have been working with melatonin in the United States, their work shows that beyond, when we get into the pharmacological dose ranges, there is saturation at the receptor level. There really shouldn't be any sound reason to go with doses that high. And so what we did was we brought the doses right down to doses that were closer to the physiological range, because we thought that if we're going to set up clinical protocols for use of melatonin, we should go with a dose that isn't exceeding what is necessary. So that's how we calculated it based on, and we've looked at dose response studies where people have done dose response studies in humans, and we find very little evidence of dose response effects in humans. Now, the only exception that I have found anecdotally is in pediatric populations where high doses are used, and I am not quite sure. Pediatricians tell me that they trial systematically increasing doses and they find that higher doses are more effective. And so that's the only difference that I would probably point to. And in terms of the last question you had, the long-term effects, I haven't seen any long-term studies that have demonstrated a change in efficacy of melatonin over time.
Here, it's just about the normal regulation of melatonin in disrupted sleep or disrupted light cycles. What happens to melatonin? Do circulating levels go down? And what's the molecular linkage between light and production of melatonin? And when you give exogenous melatonin, does it suppress endogenous production?
So I'll answer the second one first. In fact, I'll go back briefly to one of my slides where I didn't have really a lot of time to... Here we are. So this shows you the red curve is the exogenous dose. The black curve is the endogenous dose, the endogenous levels. So this is the placebo condition and this is the melatonin-treated condition. So you can see, and this is the plasma levels that were recorded. So what you can see is that in this protocol, we increased and this was with 1.5 milligrams of melatonin, and you can see already we're several-fold higher than the physiological level. So there is no direct effect of the exogenous dose on endogenous production. In terms of the connection, your first part of your question, there is a well-characterized pathway, multi-synaptic pathway between the suprachiasmatic nuclei, which regulate the central regulator of circadian rhythms, and the pineal gland, which synthesizes melatonin. And we know that when the SCN is affected by light, that directly influences the timing of melatonin secretion. And we also know that exposure to light will suppress melatonin secretion as well. And so there is this direct link between, and that's why melatonin, endogenous melatonin, is used in humans as the gold standard marker of the activity of the clock. Thank you.
Right. Well, thank you very much. Okay, so it's now my pleasure to introduce to the stage Dominic Dwyer, who's Director of the Institute of Clinical Pathology and Medical Research and Director of Public Health Pathology of New South Wales Health Pathology, Westmead Hospital. Welcome.
Thank you very much for the invitation to speak, and I'd also like to acknowledge the Wurundjeri people here in this part of Australia, and I have the pleasure of working on the lands of the Barramadigal in Western Sydney. So it's not letting me open. Let's have a go. Are you able to open from up? Thank you. So look, while we're waiting for that, if it's going to work, I'm going to talk today about work undertaken with the WHO to investigate the origins of SARS-CoV-2 and outline not so much the science of it, although that is clearly fascinating and certainly what I'm trained in, but also to highlight some of the issues that arose with that work that are... let me just say, sorry, you're doing that. The issues that arose that I think are relevant to the academy, particularly in terms of dealing with the media and the sort of anti-science sentiment that's been problematic in recent years. So while waiting for this, thank you. While waiting for the slides, it was on the 30th of December 2019 that ProMed mentioned that there was a. Do you need? Oh, you got it already. Beauty. Okay. I'll have a yeah. Okay. Thank you. And if you could make that. Right, okay, so let me just get on with this. Are you progressing? Nope, let me try from this. Are you able to move it? Try the mouse. I tried that. Why don't... Okay, so while we're waiting for an expert to fix it, thanks. So ProMed alerted to the outbreak of SARS-CoV-2 in the end of December, and now we're in the situation where we've had about over 600 million cases worldwide. Thank you. Is that now working? Beauty. Yeah. With that. So we're now experienced a series of waves, which is typical of any pandemic, occurring over the last 2 years since that first alert back in 2019. And as I said, well over 600 million cases and well over 6 million deaths. Remembering, of course, that this is a gross underestimate of the number of cases. And even in Australia, with a sophisticated laboratory testing system, we've probably got at least threefold more cases than have been officially recorded. So the WHO clearly needed to get in on the act on trying to sort out the origins of this. And so the World Health Assembly requested WHO and China to work together to investigate the origins. And there were two parts to this, and this is important. Phase one was really about reviewing the early data, trying to get an understanding of where had the virus come from, what was the virus, what were the initial epidemiologic features and so on. And the second part, which is actually the most important part, was about what sort of studies do you need to develop to take this sort of work further? Because obviously understanding the origins of disease are crucial for maintaining or producing interventions. So there were 10 of us that went from around the world, and we formed into 3 groups, although we went across all groups. So there was one group looking at the animals and the environment that those animals were kept in, both within Wuhan, where the outbreak started, but also elsewhere. Another group looked at some of the very early sequences generated as part of those early cases. And the third group looked at the sort of epidemiologic features of the outbreak itself. Now, this is, the other point I wanted to make too is that this investigation was about understanding the origins of the infection. It wasn't about assessing the responses to the infection. But unfortunately, the political approach, as you're all well aware, really became a blame game to try and, for politicians, in my opinion, to hide their own responses in their own countries to the epidemic. So this really influenced the media too. So our role was really that lower red bar there, which looked at the very early cases in December 2019. And even though so the important feature from this graph is that not all the cases were associated with the wet market in downtown Wuhan, the Huanan wet market, but most of them were. Now, we're not supposed to be talking about responses, but if you wanted to see a critical error in responses to this outbreak, it's highlighted with those asterisks, so that it was known in the last days of December 2019 that there was a novel coronavirus being sequenced and was available. So this was known, but the Chinese CDC didn't release the gene sequence of that virus till about 12 days later, and that was only because investigators at Sydney Uni and another university in a different city in China deliberately released the sequence, because having the sequence is the key to developing diagnostic testing, developing vaccines, understanding origins and so on. But we're not really talking about responses. So I don't have time, obviously, to go into what was pretty complex science, but this is some pictures from the Huanan market, and you could not script a better place to have a pandemic originate. You have this is about a 2-hectare site. It's really very, very big. Dark, narrow corridors, open drains, cages and pits in the middle for animals to be kept and slaughtered, and on the right-hand side, sleeping quarters in direct contact with where the animals were slaughtered and so on. And then, of course, you go outside, and this marketplace sits right in the middle of very dense population, typical of so many modern Chinese cities, with about 1,000 people living in each of those individual buildings. We also visited the Wuhan Institute of Virology, and this is one of the world's famous laboratories for investigating bat coronaviruses. And this all arose after the SARS outbreak in 2003. And the point of that is that, just as a reminder, it took about 14 years before the origins of SARS, the original SARS, was identified in bats. And that's just to highlight that these investigations can be very slow and painstaking. Anyway, we reviewed that. We weren't charged with doing a forensic examination of the laboratory, but this has become very much a focus of a lot of the politics and so on about countries accusing deliberate or accidental leaks of from the laboratory and so on. And plenty of scientists and so on have said the same thing. Mostly people who don't understand how laboratories work and how laboratories leaks actually occur. They do occur, they're rare, but I don't believe that was the case here. Now, there's a whole lot of work that's happened since we were there a year ago, and I'll just highlight two. One is that originally it was said that there were no wild animals in the market, no illegal animals in the market, and that's because after SARS in 2003, that trade was deemed illegal, but clearly hadn't been shut down. And this is a publication from Chinese and Canadian researchers that had pictures of that Huanan market with these wild animals in situ. So clearly the animals were there in the market, and this is in November 2019. And then the second thing is, getting back to what I was saying about phase 2 extended investigations of data. So this is work that came out in Science just a couple of months ago, where they went back and looked at the data of the cases that we'd looked at, and they found that all of the cases, those first 172 cases, were essentially located geospatially around the market. Okay, it's not cause and effect, but it's pretty good evidence. And this is in a city with a population of 12 million. They also looked at the cases that had been not linked epidemiologically to the market, but in fact found that those cases were also very closely linked geospatially to the market. So the market, and there's a lot of other evidence as well, clearly was the epicentre of the outbreak. And then, of course, you end up with this sort of scenario that we had said, and I think the evidence is all in favour of this, that it started in bats, that it went from bats into some sort of intermediate animal. That intermediate animal or animals hasn't actually been identified. These sorts of cross-species transmission don't happen as one virus across into one person. And in fact, there's very early genetic evidence of the sequences we looked at that show that there were probably a couple of introductions into that market. But one particular SARS-CoV-2 took off, if you like, and spread worldwide. Spread around the market, around the city, and then, of course, human behaviour, travel, and so on, then generates the pandemic around the world. So, but the points I wanted to make now were really about the fact that the sort of politics around this and the argy-bargy between countries, between, say, President Trump and China and even Scott Morrison and China and others, all that did was make people less likely to allow cooperation to occur. And these sorts of origin studies can only be done if there's extensive collaboration and cooperation between different parts of the One Health environment, be it human, animal, environment, and so on. And so we were concerned enough to write to the leadership of WHO for what that's worth, and also to the scientific literature to say that these sorts of arguments were doing nothing to help understand what was going on, because the key work here is going back and look at the animals and the people that look after those animals, bring them to market and so on. And the longer you leave it, the harder it is to actually do. The other thing I just wanted to point out too was then some of the responses, and this is what I wanted to, I guess, flag to the Academy, but how do you deal now with attacks on scientists and anti-science sentiment and so on that's making debate difficult. So these are some of the emails that I've received, and I've spared your blushes by not putting the ones with rude words in them, because there are plenty of those, and I still get these. 2 of the 10 people on the group have had death threats and have had to have guards and so on, and I've had to seek advice from agencies in Australia that I didn't actually know existed until I needed help. So this sort of thing then is, it's not just about the origin story, although that was clearly a focus, but it's also about vaccination and COVID, climate change and a whole range of other things. And in fact, now there's starting to be some commentary about this in the literature, and in fact an Australian survey that was done on this, pointing out the difficulties that scientists have had in not just COVID, but in vaccination, climate change and so on, as I mentioned. So in finishing up, I just want to say that these origins investigations are difficult to do and often take a long, long time. The standard sort of version of how this occurred has stood up. And this, in fact, indeed is typical of just about every virus that's emerged in the last 50 years anyway. But the important thing is, in these sorts of episodes, is not to downplay or dismiss the other hypotheses, like laboratory leaks or like frozen food and a whole range of other things. But to have them sit there, that if evidence comes forward that supports it, fine, off you go and do the work. But if the evidence doesn't come up, and it hasn't come up with the laboratory leak or the other ideas, then you just leave it sitting there. And just to make the last point, that science, politics and the media are clearly difficult bedfellows, and I think we probably have a responsibility as an organisation to try and understand why this is proving so difficult. And even though melatonin clearly works, for those insomniacs, if you want to read the many hundreds of pages of report, that'll do the job too. So thank you very much.
Well, thank you. We may have time for one question. Unfortunately, the technology didn't help. There is a question. Yes, there's one here. Thank you.
Yeah, great work, Dominic. I like your science, politics and bedfellows comment. During COVID, there was sort of a temptation of anyone that held a pipette to offer an opinion about COVID science and what everyone should do. Do you think that was sort of helpful, or how would, in retrospect, what would be the way to handle that?
Yeah, look, thanks, Stephen. I mean, you didn't have to hold a pipette to have an opinion, let me tell you. Look, I think it's really difficult. I can only go on the New South Wales ministry approach here, and perhaps on the Commonwealth approach. I don't know in Victoria. But one of the difficulties is, of course, that ministries of health are naturally risk averse, meaning that when something comes up that's complicated, they want to sit on it, work out strategies for answering it and so on. And they miss the chance to engage groups of people that would help them in their messaging. So the media would go to the ministry and say, can you get someone to come and talk to us? And they say, we want to know what are you asking and all that sort of stuff. So then the media would naturally go to people who would answer the phone and give an opinion, because they weren't in the public sector or whatever. So I think departments of health miss a chance to get a cohort of people to help them with their messaging that weren't obviously Department of Health people. And that applies not just to things like origins, but mask use, relaxing isolation, all of those sorts of things that everyone agonises about. So that's one particular approach. I think there is also then the role of organisations, and certainly the medical research group and I think this academy and so on have a role in doing that as well. But the media, as we were discussing before, the media like controversy, they like a fight, and so that's always hard to manage as well. But anyway.
Well, thank you very much. So now it's time for Emma Kowal. And Emma is an anthropologist, a professor of anthropology at Deakin University. So I'm really interested to see what you've got to say. Thank you. There you go. Got it up for you.
Beautiful. Thank you so much. And I would also like to acknowledge the traditional owners, the Wurundjeri people, who are the custodians of the land that we are living on and have never ceded their sovereignty. And thank you. Potentially, I'm slightly premature putting the Academy of Health and Medical Science up there. Maybe it's not till this evening, but very, very proud to be part of this. Thank you to everyone who nominated me. And I just want to start, because I've probably got potentially an unusual collection of letters with that BA. So I have had an unusual medical career, and I'll just tell you because I think it's wonderful to have diverse medical graduates here in the Academy. And it's really been guided by a desire to have a contribution to improving Indigenous health using the methods and theories of the social sciences. So when I did medicine, I also did a anthropology degree with fieldwork for my honours in medical anthropology in Papua New Guinea. And after I graduated in 2000, I headed straight to Darwin to work in Aboriginal health because I was an activist, probably still am a bit of an activist. And I worked at the Royal Darwin, at the Aboriginal Medical Service, but kind of quickly moved into public health, working at the Menzies School of Health Research and what is now the Doherty Institute, and eventually did a PhD in anthropology, but within Aboriginal health. And what I'll talk about today is one part of my work, which has been on the ethical, social and cultural implications of genomics for Aboriginal and Torres Strait Islander people, and an effort to build an Indigenous-led genomics in Australia. And I do want to just warn that there are images of Aboriginal and Torres Strait Islander people who have passed away in this presentation. So I want to start in Pineapple Creek in 1963, which is in between Fitzroy Crossing and Halls Creek. And I don't know the name of the man on the right, but the man on the left is Bob Kirk, who was a British geneticist who moved to UWA in the 50s to start up the Department of Biology. And he was a snail geneticist, but soon he realised there was a great opportunity in taking samples from Aboriginal people in the north of Western Australia for genetic research. And he was part of what was called the International Biological Program in the 1960s, which aimed to take stock of the biosphere and had one of its 7 arms was human adaptability. And Bob Kirk was an important contributor to the Australian arm of that international project by taking blood samples from Aboriginal people. So this is in Warburton, at the Warburton Hospital. One of my other interests, or parallel interests, is in what's called science and technology studies, which is the social studies of science and technology. I'm actually president of the International Society for the Social Studies of Science. And that looks at how science and technology shape the social world and how society shapes science and technology. So in the story of how so many blood samples were taken from Aboriginal people, what's really important in this picture is this mobile refrigeration device, probably with dry ice in the 50s. And so that the emergence of that technology, which was actually developed for bull sperm, another story, enabled the transport of biospecimens over really long distances. And the collection of samples from Indigenous people all around the world was rapidly utilised. So in 1967, Bob Kirk goes to ANU and he has a very state-of-the-art starch gel electrophoresis laboratory. And he brings with him samples like these ones from Aboriginal communities, and he gets a lot more of them until there are thousands of samples from over 50 communities in the top half of Australia, basically, which are part of a much larger collection of 400,000 samples, mostly from the Asia-Pacific region. So these were used for genetic research up until the 1990s, into the early days of direct DNA methods, and there were some samples that had DNA extractions done. So in the 1990s, there was another big international genome project, well, the Human Genome Project, we all know about that one, but there was an accompaniment to that, which was in many ways the sequel to the International Biological Program that I mentioned from the 60s. And that was the Human Genome Diversity Project, which was run by population geneticists and pretty quickly fell afoul of Indigenous groups who by the 90s had quite a bit more political power and international organisation than in the 60s. So this is actually from the publication of the Australian-New Zealand Association for the Advancement of Science, called Search. I don't know if anyone used to get that, but this is back from April 1994. And there was quite a bit of media coverage at that time about Aboriginal people not wanting to participate in the Human Genome Diversity Project. So this was very big news at the time, and certainly a bad news story for science in Australia and internationally. And at this time, the samples of Bob Kirk, which are at ANU, I should have said in the John Curtin School of Medical Research, stopped being used. There was a decision to close down the collection for study. But it just stayed there, that no one really knew what to do with it. So I had been working on the question of genomics for a few years by the time we got to 2010, nearly 12 years ago exactly, when I called cold the Professor of Genetics, Simon E. Steele, who was the custodian of the samples, and that began 7 years of pretty intense work to develop, to try and find a way to manage these samples. So we brought together an Indigenous consultative committee from probably many people that you recognise and some members of this academy. And what came out of that was the first Indigenous governed genome facility in the world. And this is at the launch with a few of the board members and two of Bob Kirk's two daughters. So I've been involved I was involved in many parts of setting this up, including developing some information resources for engaging with communities where samples had been taken from, and having conversations about consent for samples being retained in the collection and to be sequenced. So this has gone on to be, gone on to wonderful things. And ANU, I should mention, continues to lead in Indigenous genomics in partnership with the Telethon Kids Institute and Alex Brown, who is also a member of the Academy, is just recently become the first Professor of Indigenous Genomics in the world. So pretty interesting stuff. Now, just the very last thing in my last 2 minutes is to talk about Indigenous workforce. So it's wonderful to have this really Indigenous-led engagement with genomics, but we also need, Alex is obviously amazing, but we need more of him. So since we can't clone him, we need more people in Indigenous genomics, more Indigenous people. And so I've been involved in what's called SING, the Summer Internship for Indigenous Peoples in Genomics. This started in the US in 2011 and as SING US, and then there's been programs in Canada, in Aotearoa, New Zealand, now in Australia, and most recently in Mexico. And it's a training program for Indigenous people to be exposed to all aspects of genomics, from lab work. So I got to extract DNA for the first time, which was really cool. It is just like cooking, I've got to say, not going to lie. So this is our recent workshop in Adelaide, and we also, engage with cultural activities as part of that, and have the vast majority of the faculty are now Indigenous. So I just want to say thanks to all of the organisations. And that it's supported a lot by Australian Genomics, and I really want to thank them for that. And thank you for listening.
Well, thank you very much. Now we do have time for your questions.
10 seconds under.
Yes, well done. Just wanted to look. What have you done scientifically, though? I mean, that was very interesting. But I'm just dying to know what your science has been.
Well, I'm not a scientist, so that's easy to answer.
Right. Okay, that'll do it.
I'm a
Oh,
social
social scientist.
So yeah, I have a lot of publications which I haven't talked about here, but you can look me up on Academia, which is the platform of choice for social scientists to share their publications. So I've really analysed what it means for Indigenous people, and for I'm really interested in biological difference and the stakes of creating biological knowledge about other people, particularly people who have been marginalised. So this is a practice with massive history and a very difficult history, and I tend to really get interested in things that are difficult. So genomics in Indigenous people, that seemed like a really complex and difficult area. When I wrote my first postdoc application in 2006, there was basically no genomics happening of any kind in Aboriginal communities, and it just seemed like a really outrageous area to try and look at. But I knew that it was eventually going to be a thing, that genomics would eventually be used in Aboriginal health and be important, and also because of the issues of inclusion and all of the resources that were going to go into genomics and precision medicine. So I wanted to try and make sure that in Australia genomics was not harmful to Aboriginal people, and hopefully I've contributed to that. But not a scientist.
Okay. Yes, there is a question down here, thanks.
Hi, Emma Ainsley from University of Sydney. Congratulations on being appointed as a fellow, and thanks for your presentation. I'm also not a scientist in the way that you're not a scientist. So
Start a support group.
yeah, I think I was just going to ask you, I guess your point then actually just kind of really helped me articulate my question better around avoiding harm. And initiatives like Sing, I think, are going to kind of help in that way. But how can we use genomics well in Indigenous populations? And I guess a second point is how can you and I, as non-Indigenous folks, help? Like, how can we help while not taking over, I guess? So I want to make sure that I'm the best ally that I can be while not taking ownership where it's inappropriate, I guess. That's something I question for my own work.
Yeah, no, absolutely. And what are you doing here is a question that, people should ask me all the time. And sometimes they do. Sometimes they're probably just polite, which is okay as well. I don't mind politeness. But yeah, I mean, I've worked in Aboriginal health for all of my career for well over 20 years. So, and previous to that was at university, was really engaging with Aboriginal activists and, trying to learn from them. And so one of my origin stories is Gary Foley, who's a very important legendary living legend at Melbourne Uni, who he told all of us student activists, just focus on your own people, don't try and help Aboriginal people. So that's kind of been what I've tried to do in just trying to make healthcare less racist, to try and promote Aboriginal control of every area of health and medicine.
That's probably just about it. But Emma, I hope you don't mind if I think of you as a social scientist. As actually a scientist. I mean, I didn't really make that distinction in my own mind. And I trust that you will accept that.
Yeah, sorry, I might have self-identified myself out of where I didn't need to.
Great. Well, thanks very much, Emma. That was a wonderful talk. Okay, so the last speaker in this session is Laura Mackay, And Laura is professor and laboratory head at the Doherty Institute. I'm sure we're going to hear more about which laboratory that is. There you are. Great, thanks, welcome.
Thank you so much. Sorry, I have a support team for my talk. Great, and while that starts, I would like to thank the Academy for the opportunity to speak to you today about some of our work. My name's Laura, I'm an immunologist at the Doherty Institute, and we work on memory T cells. So it's a credible honour for this evening to be, I share Emma's sentiments of being so proud to be inducted into this really prestigious group later today. So we're interested in cellular immune responses, and particularly those that occur in tissues. So we know that immunological memory is when our body can remember something they've seen before. This is also what we want to induce with new vaccines. So here's this image here is infected skin, and you can see here in the outermost layer are green cell. These green cells are our memory T cells that are embedded within tissues. They can directly recognize and kill pathogens that enter. So typically, immunologists have studied T cells as they're induced. These are T cells in the thymus and the spleen, the lymph node. This is where immune responses are initiated. Where we sample immune responses for the most part is in the blood. But what we found, and now many groups around the world are working on this, is that it's really these embedded T cells that do a lot of the heavy lifting when it comes to protecting against infection. So generating good immune responses is really the basis of vaccination. We want to generate and maintain long-term memory cells that can protect against pathogen exposure. And so here's one of the best vaccines of all time, smallpox, which eradicated this infection. You can see it induces incredible antibodies that last for years after vaccination, and also a really, really good T cell response. And for most of the vaccines where we don't have, most of the diseases, where we don't have good vaccines, it's those where we've failed to induce good T cell memory. And perhaps we won't need to keep having COVID boosters if we can make a vaccine that induces T cell memory, as the vaccines so far are targeting our antibody responses. So what do we know about memory T cells? They circulate around the body, and so when we get an infection or we immunize, these T cells will be migrating all around the body, and they'll enter the infected site to try and kill infected cells and clear the infection. Now, if you get an infection, of course, that takes time unless the T cells happen to be wandering through the tissues at that time. And generally, the immunological community thought that if we take some tissue, like the gut or the skin, you see a CD8 T cell in there, that's a memory T cell that's just passing through. This view really changed around 2009, that actually the T cells within tissues are distinct. So tissue was transplanted, and it be found that cells of donor origin were actually embedded in the tissues. So they weren't in conjunction with the circulation, and they were residing there. And this is what a lot of vaccines have failed to induce, largely because we didn't know what we were looking for. So fast forward to 2022, it's now well accepted that tissue-resident memory T cells, or TRM cells, are a distinct memory T cell subset. They are distinct in terms of phenotype, development, and functionality, which we've been working on for many years, and their cardinal feature is that they don't recirculate. Of course, this has implications for tissue transplantation, because you wipe your T cells. If you wipe your T cells out, you're carrying across tissue with T cells with donor origin in. And something that we've been working on is the fact that these T cells are special. So properties that are true of T cells in the blood are not the same for those T cells in the tissue. For example, they're radioresistant when they're in tissues, they're not in the blood. So they've been identified pretty much in all tissues across the human body now. And why should we care? Are we just immunologists trying to put things in boxes because this is what my breed tends to do when we like to ascribe different CD numbers, and you may all remember this. Why should we care? So it's been shown by many groups now that these T cells are naturally how our body protects against reinfection. So it's been associated in a lot of mouse models that TRMs provide protection against reinfection, particularly against pathogens that enter via mucosal sites. In humans, it's been shown that a high number of T cells with this phenotype correlate with subclinical herpes virus reactivation. This means where you don't see overt disease, elite controllers of HIV, and also improved outcomes against cancer. And so it's thought that vaccines which can be geared to induce this T cell subset specifically will result in superior immune protection. And just a sort of example here in mice from us is, say you take a naive animal, you boost a lot of antigen-specific circulating T cells, or you immunize to induce a lot of resident memory T cells. I'm going to spare you the details, but we've got various genetic modalities so we can have more of one and not the other and eliminate the other completely to see what's going on. And we can do this because these express distinct surface molecules. And you can see when we challenge with a virus like herpes, which has this classic zoster-form lesion, it's only when you embed TRM cells are you able to protect against disease. So one of the ways to illustrate this finding will be something like this. So a complication which I'm not going to mention is that also these TRMs, because they're in tissues, they have distinct functions and properties. They can actually kick off responses a bit faster. But if you think about a virus that's entering a peripheral site, if you've got to wait for circulating T cells to wander into the tissue, there's going to be a temporal delay. And this will give the virus the upper hand. It will be able to take hold and disseminate. And in that example of herpes, come back and give you that zoster-form lesion. If you're able to embed resident memory T cells directly in the mucosa, this might offer you a rapid control and containment of the virus. And more recently, we've shown that also in states of melanoma, for example, or solid tumors, if you can embed resident memory T cells within the epithelial layer, that's able to keep the melanoma in check and keep that cancer immune equilibrium. If you then specifically eliminate these T cells, the melanoma grows out. So it's now well appreciated, at least by immunologists, that resident memory T cells are central to many aspects of cancer and infection, and boosting these cells in these contexts is a focus of new immunotherapies. And also there's a flip side of having a lot of T cells that are ready to go in tissues. They've now been associated with various pathologies, particularly those in skin. And you may think those sorts of disorders which always come back at the same place at the same time, like fixed drug eruption or vitiligo, psoriasis, a lot of these are a thought, at least, and there's a lot of ongoing research thought to be mediated by this TRM cell subset, which don't move within the tissue once they're established, and they don't even move laterally. They're very contained within the tissue. So to try and make new TRM-based therapeutics, the starting point is, of course, to really understand the biology, and this has really been our focus over the past few years. So we showed back in 2013 that these T cells were transcriptionally distinct. And really this was what was needed to get a lot of immunologists on board to show that they were different from memory T cells in the circulation. So you can see you've got T cells clustering up here in this quadrant on this PCA plot, just showing the transcriptional similarities. So these are all different circulating T cell subsets clustering together, and then TRMs induced by different infections within different tissues, they have shared features. Once we had a handle on the genes that made a TRM cell, that was what opened the box for us to be able to start modulating these genes to induce this population. So the identification of these of markers that could be used to look for these cells then enabled us to make the jump from mice to humans and identify these T cells in humans and show that they were there too. And it led to work showing that the TRMs in humans have a common transcriptional signature which overlays with that in mouse, as compared to human T cells in the blood, which was really remarkable, and also allowed the identification of these T cells in tumours. And together with colleagues at the Peter Mac, we're able to show that in triple-negative breast cancer, for example, those patients that have high numbers of TRM cells tend to do better. And this has been echoed in many different solid cancers. So we're interested in how TRM cells develop. It's not a default process where T cells wander into the tissue and decide one day they're going to live there. It's a very coordinated process with many transcriptional regulators and suppressors acting, and this is what we've really been working on. There seems to be a distinct precursor which can be manipulated and say, if you think about how we want a great immunization for the mucosa, what's the route of immunization? Is it going to induce these cells? What's the adjuvant that you're going to use, and how does it affect cells that are going to go go on and live within the tissue. So there are certain transcriptional regulators that were key, as I mentioned, and so this is the path to residency, and this is the path to become a recirculating memory T cell. And you can see as one example, their surface would be littered with various receptors such as S1PR1 and 5, which allow tissue egress. So these have to be shut down for these T cells to settle. So over the years, we've been working out these genes and how to really change this balance of becoming more likely to give rise to a tissue resident cell versus one that circulates in the blood. And also working out all the different environmental cues and the type of cells that these cells talk to keep them happy long-term within the skin. And we know that these T cells can last for years. There's been examples of, say, facial transplants where cells of donor origin with that HLA type have been carried across, found 10 years later. So to embed these cells, you need specific signals, and some of our work now is kind of moving and informing different translation, particularly in the realm of pathology, where it's shown by treating with local IL-15 can knock out these T cells, and that's been now trialed in vitiligo patients because these cells are so very sensitive to this cytokine as opposed to those in the circulation. So some of the takeaway messages to leave you with today is the need to evaluate tissue T cell responses because the blood may fail to capture what's really going on in the tissue. And of course, you would want a great vaccine to induce the tissue immunity, so blood might not be the right readout unless you can look for the type of cell that might be geared to become one of these tissue resident cells. These cells provide local immunity against infection and tumors, and certainly we and others are working towards driving new therapeutics that will modulate TRM cells, beneficial for new vaccines, immunotherapies for solid cancer, and we're doing this in collaboration with Pfizer, and to eliminate these cells in terms of pathologies. So just to thank everybody involved in the work. This is my team over at the Doherty Institute, which is just across the road. These guys are the people who do all the work in the laboratory, and so credit is 100% to them. All of our collaborators at the University of Melbourne and elsewhere, our funders that allow us to continue doing our work, and thank you again so much for the invitation to speak today.
Well, thank you very much. Oh, we've got a question straight away, but I can't help asking my question first. You know, young children, being an ENT surgeon, I see mucosal disease in them all the time. And is kind of childcare TRM school for the children? Is this what happens in childhood development?
Well, actually, it's something that you might expect, and we're only sort of now we can find the cells we can do this, is it's shown that your TRM populations increase with age, and that makes total sense, that as you get older, you get more and more resident memory T cells, and we've actually found that one of the transcriptional regulators, which makes you want to become a TRM, is actually low in infants. So resident memory is really important for persisting infections in older patients in particular.
Interesting. Thank you. Now the real serious questions. Yes, I think one here. Thank you.
Congratulations. Mine is also an age-related question as I compare my age relative to yours, also just being admitted to the fellowship. Two of the diseases used as examples, zoster and malignancy, are both more common in older people, actually. And I was wondering if there might be, maybe there's an age-related peak after which there's any decline related to senescence for those diseases to be so obviously more common in older people.
Yeah, so TRMs wane with time, and this is something that we're trying to get a handle on how to keep them, how to maintain them in the tissue, and how things that certain therapies that are being used might be having adverse effect on these cells that we don't understand. Their fate seems to be terminally fixed, but their survival is also, it's very, very long term in the skin, but less so in other tissues. We don't know why, say, this is why the correlations are so striking. With people that keep getting recurrent bouts of simplex, for example, and it's taken really amazing teams to do this work, have shown that these people have high number of TRM cells, and those people who keep getting overt disease have low numbers, and what is regulating that balance in people is something we don't understand.
Yeah, that was fascinating. Could you Two questions. One is, are these TRM cells relevant to COVID-19 responsiveness, whether you get very sick or not? And the second one is about the brain. How important are they in brain disease and which diseases?
Yeah, so in the case of COVID, there's a huge push by people that work on this cell type to say for new generation vaccines, which would also induce T-cell immunity, the intranasal route would be really important because it induces these cells. With collaborators, something that we've been working on is actually you can have a lot of cross-protection from COVID by from just a common cold. There is a cross-reactive sort of T-cell specificity, which is also very protective against COVID. And we find that they're actually in high quantities in the human lung. So whether that's correlated with why some people do better and what, I'm purely speculating. I don't really work on COVID. I'm sure Steve Kent is thinking, what is Laura talking about? I know way more, and I'm sure he does. So we can speak to him later. With regards to TRM cells in the brain, they've been identified in mice and they've been shown to protect against cerebral malaria. In human patients, I don't think anything's been done so far.
Thanks.
We probably don't have time for another question. So thank you very much, Laura. So look, I'd like us now to thank all the speakers for some great talks, and thank you.